right, well, welcome. Good morning, good afternoon. My name is Raluca, and I'm a proud staff member of the Protein Society. I'd like to first and foremost introduce Shannon, who is my co-pilot here.
Hello.
And we'd like to welcome you to our 37th webinar, organized by Justin Benesch, and with the title Mass Photometry. We are very, very proud and glad to have you here. We also have an upcoming webinar on careers in industry that we invite you to register for. Protein Society webinars are always free. And I also wanted to mention that Pride and Joy at the Protein Society is our annual meeting. We will take place in Boston in July, and we hope to see you there. And we encourage you to submit your abstract for what we hope will be close to 100 travel awards. And there are also open talks in every session. And I'm very proud that the Protein Society has not raised its registration, membership fee, or any other fees since 2019. So please take a look at our website to learn more. And last but not least, our Pride and Joy, our protein science journal, with an incredible team led by Dr. John Kirian as editor-in-chief, and senior editors Nir Bental, Zengi Chang, Eitzaber Lopez Cortajarena, and Lynn Camerlin. This is a community journal just for you, our protein scientists. And again, thank you for joining. I am now going to turn it over to Dr. Benesch, who will walk us through. And thank you for organizing this webinar, and thank you for all of our speakers.
Great. Thank you so much, Raluca, for organizing this, and Shannon too. Right, it's been great to have you steer the ship, and it's a real pleasure to host this webinar. So thank you, of course, to Sarah, George, and Maria for being our speakers today, who I hope are really going to highlight some of the interesting applications that mass photometry can help explore. So just as sort of by way of brief introduction, mass photometry was a technology that was introduced now, almost 8 years ago, and I was happy to be involved with that. And it sort of stemmed from data such as the stuff that you see on the right-hand side, which are movies of individual single molecules that are unlabeled, that are being visualized on a microscope, and visualized in a way that we coined at the time as being sort of a quantitative mass imaging, and is what we know now as mass photometry. I'm not going to spend too much time dwelling on the technology in a sense, but I think what is really relevant is that even though you get those kinds of movies, which are visually super appealing, it's the fact that you can actually extract information from them that is quantitative, namely with respect to a mass axis, and on the y-axis a counts, in other words, a proxy for concentration, that allows you to actually start to think about things from a biophysical perspective, right? So the video I just showed you was of a protein that actually exists in multiple different ligomeric states, which you can see in the resulting mass spectrum. And each of those ligomeric states is only separated by less than 50 kilodaltons, actually. So mass photometry then is a way of actually obtaining mass spectra, not through mass spectrometry, but through an imaging technology instead. As I said, we're not going to spend a lot of time today talking about the ins and outs of the specifics of how to do the experiments and so on. And I do want to just point you towards a couple of resources that I think might be helpful for those of you who wish to do these experiments or want additional tips on the experiments that you're already doing. I'll start off at the bottom, and I've just sort of picked out 3 relatively recent protocol articles that I think are worth checking out to understand how to perhaps reach sort of best practice in these experiments. And I'll also point you to the website of Refine. So Refine is a company that I should declare I'm a founder and shareholder of, and still work as a consultant for, that actually sells mass photometry instrumentation. Their website does also have handbooks and lots of protocols that I think might be useful. I should say that I'm not representing Refine in any way on the call today. I'm just associated with the company, but they're not sponsoring or in any other way supporting this particular webinar. The webinar that we have then, so I'm going to start and give a little bit of an introduction to the technology and some more recent uses of the technology. Then I'm really delighted that we have a PhD talk. Sarah Delor from the Biomass Spectrometry Laboratory in Strasbourg will give the talk after me. We'll be talking about membrane protein characterization using mass photometry. Georg Hochberg from the University of Marburg will give a talk after that, talking about using mass photometry in the context of understanding the evolution of a metabolic enzyme. And Maria Spies will give the last of our talks, showing how to combine mass photometry with single molecule fluorescence imaging to study nucleic acids and proteins and the complexes they form. So what we've tried to piece together here is a program that will contextualize mass photometry as part of that sort of protein science toolkit that, of course, the Protein Society has been so good at promoting. So that's the program. We do invite you, please, to submit questions that you might have for consideration and answering. Those that we can't answer live on the webinar, we will address in the chat to you. And we do have time at the end of the overall webinar to have a little bit of discussion amongst ourselves, where we may pick up on some of the themes that have been raised by those questions. So please do submit those questions, and we can try and engage with you. So without further ado, then I'll start off and I'll give a relatively short presentation that I hope provides some context for the subsequent talks that you will hear. And I'll start off, of course, by thanking my brilliant group, and they're such a pleasure to work with, and I'm glad to be able to show some of their results today. So what is mass photometry? Where does it come from? How does it work, right? So I'm going to give a sort of slightly high-level view and give you one of these videos again, where you can see these black dots appearing when we're visualizing and videoing a microscope cover slide. So all these black dots correspond to a molecule that has landed and interacted with the glass surface of that cover slip. And so how does that happen without these molecules being labeled? Well, the concept really comes down to interferometric light scattering. So we're shining light from the bottom of the glass onto the bottom of the glass cover slip. Some of that light is reflected back, which I should get my laser pointer. So some of that light gets reflected back, but some of the light is also scattered by molecules that are close to bound to that surface. And as the interference between the scattered light and the reflected light is the signal that we actually measure. And so we're videoing that interferometric signal. And you might imagine that that interferometric signal is going to depend on the size or the contribution from the scattered light, right? And so the larger the scattered light contribution, the more we might see contrast in the video. So let me address one question that often comes up, which is, why do you see this sort of flickering, sort of dots appearing and disappearing? And that comes because what we're actually videoing is a change at the interface, right? So we visualize what is going on at the interface in a sort of time-dependent manner. So if we look here on this slide, you might imagine one molecule is bound to the glass surface, and a moment in time that gives a lot of contrast. But as we think about over time, that becomes part of the background, right? Because it's no longer a current thing that's happening. Whereas a molecule that's further away that moves towards the interface gives very little signal, but as it comes to interfaces, hits the surface and starts to be imaged, we then see the signal rise up to a maximum and decrease, right? And so what we end up getting actually is, over time, we see increases in contrast and decreases in contrast, which are really a function of that molecule not just gliding with the surface, but also our videoing. And so what we do to extract the mass, or in the first instance, we extract the contrast, which is just a histogram up the maximum contrast for all of the events that we see. And so if we think about that, I'm going to give you some examples for the protein I showed you before. This actually is just it's just plain old boring BSA. And BSA, as some of you will know, has a small population of dimers and trimers and tetramers as well. And so these are representative images of those monomers, dimers, tetramers, and tetramers. And so you can see the tetramer gives a much darker dot, so more contrast, because there is more of that protein there. It scatters more light, and it gives rise to higher or large interferometric signal. So those are representative snapshots. If we then look across the entire movie and histogram all of those up, we can start to obtain a contrast histogram, which is here in gray, right? So the y-axis here is time. Many events fall with a contrast around about 0.5%, fewer, but still a lot, with about double that, fewer with triple and fewer with quadruple. So what was obvious here is that the spacing here is pretty regular. And that fits with this concept of BSA having multiple oligomeric states. And at the time of making these measurements, we wondered whether you would actually see this linearity or apparent linearity over a range of different proteins that may have different compositions and different structures and shapes and so on. And even then in 2018, where there's only whatever 10 or so data points on this graph, you can see that the expected mass based on the sequence of the particular protein actually correlated linearly with the contrast that was measured. So in principle, what that meant is that if you have a protein of unknown mass, by measuring the contrast and applying a suitable calibration, you could extract the mass of that protein. And the error was on the order of a couple of percent. Perhaps unsurprisingly, there were some slightly larger deviations at the very low masses. And with today's instrumentation, these errors are a little bit better, or they are better than they were then, and they remain, or the low error persists to lower masses. So that's the sort of the concept behind the methodology. So what I want to talk about as sort of a new area of science for us is to think a little bit more closely about what can be done with these experiments, right? So whereas just by measuring molecules binding to the surface and giving a mass spectrum, and you will see nice examples from our three subsequent speakers on that, I want to think about what might happen, not just if a molecule binds to a surface, but also if it undergoes subsequent behavior from that surface. So I'll try and play that movie again. But if you watch this movie, you will see some of the dots in here are actually white rather than black, right? They're actually equivalently, how do you say, they're just as white as the other ones are black. So the contrast is the same, but opposite in sign. And so what that can be ascribed to is, whereas a positive contrast can arise from a molecule of a certain mass landing on the surface and giving us a certain signal, if that molecule subsequently were to unbind from the surface, it would give the same size of signal, but with opposite sign, right? So illustrate that here with this white signal. But what about if the molecule didn't unbind in its entirety, but in fact actually just lost some mass, through losing a subunit or losing a set of subunits? Well, then you would also expect to see white dots, but those white dots would not be of the same contrast as if the whole molecule had come off. They would be less bright, effectively. So it's almost like doing the mass photometry experiment that you're familiar with in reverse, so the inverse of it. So we thought this would be an interesting thing to follow, because we thought what we might be able to do is link together a binding event with an unbinding or dissociation event. So let's think about it like this. Let's imagine we watch this movie and have taken a couple of frames, and we saw a binding event happen in this position on the glass surface. And sometime later, we saw an unbinding event, so a corresponding white dot appear in what seems to be the same location as where that black dot was recorded. So this is a putative unbinding of a molecule that we've seen bind. In this case, on the right-hand side, we saw a molecule bind, and sometime later we saw what appears to be a dissociation event. So a white event, but much less contrast than the unbinding event on the other side. So this is a putative dissociation. So the idea is, well, let's try and match these things up, right? Let's try and match all of the possible unbinding and dissociation events to a precursor or parent binding event. So that's actually slightly easier said than done. And that's because we're recording many, many events over the course of a video. And if I go back, you will see that the size of these events, even though they come from a very, very small molecule, the image they generate is actually much larger, right? So it's not so obvious when you're recording hundreds or thousands of events that you're going to be very successful at linking up a binding with an unbinding, or with doing that correctly. So here's a map of molecules that have attached to the surface as they bound. Those are in blue, and an equivalent map of all of those that subsequently unbound or dissociated in red. And the size of those circles is proportional to our localization precision of those events, right? So the heavier a molecule, the more contrast it gives, the better we are at localizing exactly where it bound. So it gives a smaller dot. So I can zoom in, and so the really small dots, perhaps counterintuitively, are the ones that are the heavier molecules, and the larger dots are lighter molecules. And you will see every single red circle here, or almost every single red circle here, we're able to overlap with a blue circle. In other words, we're able to pair up a dissociation or unbinding event to its likely parent binding event. So that gives us pairs and effectively allows us to do a two-dimensional experiment. So how would we expect our data to look? Well, we're now measuring the mass twice, effectively. We're measuring on the x-axis the precursor mass, so the mass, the binding mass, if you will. And on the y-axis, we may have an unbinding event that we can ascribe to that parent ion, right? So if a molecule just bound and then unbound, it would have the same mass for binding and unbinding and would land on this diagonal. However, if the molecule that unbound was only, say, a subunit of the original parent ion, then you would be in this bottom right-hand triangle where you would be probing dissociation, right? So this bottom right-hand triangle corresponds to dissociation. The upper left-hand triangle would correspond to association, something which we think should, in principle, be really very rare. And actually, things up in this triangle, we probably interpret as false positives in the pair matching that has been carried out. So how does this look like with real data? Well, here's an example. This is a hexameric enzyme. I'm not going to talk much about it because I know Georg will mention or go into some depth about this, but this is a citrate synthase from a particular organism that exists as a hexamer. And we see the molecule binding primarily the hexamer, so that's on the x-axis, and we see a lot of those hexamers losing dimers, right? So the mass of 100 kilodaltons is recorded for a molecule that comes off something that bound with 300 kilodaltons. So we can describe this or ascribe this to the hexamer being constructed of dimers, the trimer of dimers, and releasing those dimers at a certain rate. So that asked the interesting question. It was like, well, okay, why don't we ask how long it takes for a dimer to be released from a hexamer? And this is what we get. So we can extract a decay time for all of these. I think it's almost 1,000 individual dimers here that were released from the hexamers, and we obtained a decay time distribution that is essentially exponential. For the eagle-eyed amongst you, you'll note that it looks as though it's a slightly activated process, which has its own interesting biophysics with it. But we can describe a characteristic off rate to that, or off rate constant to that dimer, dimer being released from the hexamer. This is an interesting concept, and the question was, well, is this a real thing? Can we probe whether this decay time is actually reflective of the underlying biology of the protein here? And so we added the substrate and asked the same question as to whether, what is the rate constant for dissociation of the hexamer was. And what we found is that decay time distribution is substantially shifted. And in fact, if you look at the rate constants that we extract, it's roundabout, or it's pretty close to a factor of 2 slower for that dimer to be released in the case of there being substrate attached to the enzyme. So this shows us that we can actually start to probe meaningful biological interfaces and kinetics. So that protein was a hexamer. Another protein from the same family that Georg gave us to look at actually exists as an 18-mer. And this protein, therefore, gives rise to signal at 800 kilodaltons. It also exists as a hexamer and a small population of dimers and monomers as well. And so this actually is a lot of data that's combined here. And we can see this protein likes to dissociate primarily via the loss of dimers, but also via loss of monomers. And we can really see tetramers, hexamers, 8-mers, and all the way actually right up to loss of 16-mers from that 18-mer, right? So this can break at all the dimer interfaces within the oligomer. And again, here you can ask the question of like, well, what are the kinetics of these processes? And so we've just chosen 2 parts of this 2-dimensional spectrum to focus in on. We're looking to look at the loss of dimer from the 18-mer and the loss of dimer from the hexamer. And if you compare those, you can see that actually the rate constant is slower. It's a smaller number for the 18-mer than it is for the hexamer. Again, different biophysics of those interfaces in the context of which stoichiometry they reside within. And interesting enough, again, if we add substrate to this, we see both of those rate constants get depressed. The stabilization that the substrate provides is present in both the hexamer and the 18-mer states. So this is for us, I think, an interesting advance that we think we can use mass photometry not just to measure the mass of molecules once, but to measure them twice, right? To look at dissociations as well as initial parent ions. And it also leads to the question of, well, if you can just measure something happening once, can you not just keep on measuring it, right? And that's sort of logically true, right? We can keep looking at the molecule as it's on the surface and see what happens to it. And to sort of illustrate that, I'm going to take, in this case now, quite a bit larger molecule. This is an adeno-associated virus. So around about, whatever, 3.5 megadaltons or so. And this is that one of these molecules on the surface of the glass, right? And so what we're going to do now is rather than imaging the change of the glass interface, we're just going to start imaging that glass interface and ask how it changes over time, or does anything change over time? So that's the first image, and let's get the video going. We can start monitoring that molecule for seconds, tens of seconds, and record slight changes in position and record the mass the whole way through, right? And you can see that mass stays pretty rock solid at just under 3.5 megadaltons, such that, over whatever 20 seconds or so of this movie, we can then sum all the data together and obtain a pretty accurate mass for that one single molecule as it stayed bound to the surface and shifted around by a few nanometers, but more or less stayed in the same position. So that's an example then of a molecule that stayed at exactly the same mass. But when we start looking around at these AAVs, right, and for those of you who are not familiar with AAVs, they are virus particles that have 60 subunits within them. Um, and some of them we found that actually decayed in their mass, right? So started off at 3.6 megadaltons or so, but over 30 seconds actually decayed significantly in mass. They moved around a little bit. And the interesting thing is that if you take this mass trace and histogram it up, right? So histogram it up onto sort of the this x-axis, you can see peaks in this distribution, right? And our question was, well, what are those peaks separated by? And actually, if you ask that question, you can sort of plot this on a graph where you assign an arbitrary unit on the x-axis as a function of the mass, and you will see that those peaks are separated by about 60 odd kilodaltons, which happens to be very, very close to the average mass of the viral proteins that comprise the AAV. So actually what we think we're seeing here is the induced dissociation of the AAVs as they are on the surface and being heated up by the laser that drives the mass photometry experiment. So that's where I wanted to end my presentation today. I really wanted to highlight three people from my group who did all the work that I showed you today: Josh Bishop, Alex O'Shea, and Dom Saman. This work has been part of a long ongoing collaboration with Philip Kukura and his group. And the work on the citrate synthase was with Georg Hochberg, and Susumu and Tetsuo have been collaborators on the AAV work. So I'm very grateful for their willingness to work with us. And I want to thank my funders and thank you for your attention. And I'm very happy to take a question or two that George will field.
Thank you, Justin. We actually have a few questions in the chat. I'm going to start with a very simple one: what the appropriate concentration range for mass photometry is before your slide gets a bit too crowded.
So I think that question is an important one. I think the first thing I should say with this is that when we think about concentration in mass photometry experiments, we need to be thinking about the concentration of the individual particles, right, which may differ from the concentration of the number of monomers, say, right? So direct comparisons with, say, UV-Vis measurements are sometimes off by that factor. Having said that, though, the particle concentrations we like to operate in are below 100 nanomolar typically, and possibly the sweet spot is about a tenth of that. There might be slight variations depending on how well or how transiently those molecules interact with the glass surface, which is something that will vary a little bit from one protein to another.
The next question is whether this is more appropriate for globular rather than sort of unfolded proteins that have a larger hydrodynamic radius. So I guess this is about at what point we're moving out of the Rayleigh scattering regime and whether it matters if we do.
So that's an important point. So what I didn't mention, and I probably ought to have, is that we're essentially looking at molecules within a diffraction limited volume, right, which is on the order of 200 nanometers in diameter. So if your molecule is substantially smaller than that size, than that dimension, in any of its dimensions, in all of its dimensions, then you should expect it to behave as a well-behaved scatterer, and whether it's globular or linear on that scale oughtn't to have a significant impact. Now, there can be molecules that can be very extended, for instance, DNA and so on, that could start to get to those lengths, and they can start to give rise to what we can probably call slightly funky looking signals, at which point your contrast scaling with mass is not going to be reliable anymore. Now, I would say that to give you a sense of what mass range this becomes a problem at with proteins, it tends to be in the tens of megadaltons, right? So the AAVs I showed you before, which are at 3.5 megadaltons, when they're empty, and 4 or so megadaltons when they're full, they have a diameter of 25 nanometers or so. So almost an order of magnitude smaller than that diffraction limited dimension. So I think if there are other questions, maybe I'll field them at the end because I'd like to stick to time.
Yeah, I think there's just one more very related one where what happens if
Okay.
you have two domains with a long linker? Would you see that as a single particle or
Yeah,
as two individual particles? But I think it's the same answer, really.
exactly. So that'll depend on how long that linker is and the persistence length of whatever that linker is. So compare those dimensions with the ballpark 200 nanometers. Field a couple of questions from the audience. There's two which I think I'll pick out. The first was that the question says, or I'll paraphrase, that they previously thought that it was hard to use detergents in mass photometry, and it gives rise to too much noise. And how did you manage to get such beautiful data?
I suppose so. Okay, so in my experience, because I've worked on several different types of membrane proteins, some are a little bit more tricky to work with than others, and it's a work of trying to find a sweet spot in terms of, in I guess in the case of detergent concentration, to have enough concentration that the protein can be stable enough during the time of analysis without it being too much that the background noise is completely overwhelming for the instrument.
And did you have to fiddle around with different, do you have to screen lots of detergents every time you do this, or?
So for me, it was more a matter of working with the concentration. Initially, when I did without any kind of processing of the sample, I just tested concentration and see which would give me good quality data without and because also when we're working with the detergent, it can shift a little bit the surface tension of the droplets, and that can have an impact and warp the measurement of the mass. So adapt also the calibration condition to working with membrane proteins. Yeah, it took a little bit of time to be able to get this quality of data, but it's optimization that's quite easily feasible.
Well, the data is really wonderful, and there are a couple more questions for you, which I'll invite you to answer in the Q&A. But thank you so much for your talk.
Thank you very much.
And let's move on to our third talk. George, if you could share your screen. So let me introduce Georg Hochberg. He's at the University of Marburg, and is going to be talking to us today about the evolution of what you can see is a very beautiful enzyme. Thank you, Georg.
So can you hear me? I hope you can. Good. So this is a story about all the strange things you can discover if you find new ways of looking into cells. And of course, the new way of looking into cells we're here to talk about is mass photometry. And the topic of this talk is homomeric protein assemblies. So these are assemblies where you have multiple copies of the same protein make a complex. And this is especially common in enzymes, and sometimes there's a really good reason for why enzymes do this. For example, you could envisage that the active site is located between the subunits at the interface, so the assembly is necessary for activity. Or it's also often the case that these assemblies allow allosteric regulation by binding of some regulator to one subunit that changes the conformation at another. And that might only be possible if you self-assemble. But there are also very many enzymes that have one active site protemer and no known allosteric mechanisms that rely on self-assembly. So why do they still make these assemblies? And that has led some evolutionary theorists to propose that perhaps these structures kind of come and go in evolution because they're simply so easy to evolve that they can just come into existence so long as they do no harm. And I guess one piece of evidence for this theory is that the exact self-assembly state can vary quite substantially, even within particular families of proteins. And this was sort of from a few years ago where we started off. And at that time, it wasn't easy to characterize self-assembly states across large families very easily because it was very labor intensive. And that, of course, changed when we had a mass photometer available to us. So we thought we would just take a very boring enzyme family, which we thought was boring, so the citrate synthases, and they catalyzed the first reaction of the Krebs cycle. And then our plan was to go through the phylogeny of bacterial citrate synthases, where they kind of always perform the same function, purify these enzymes, and just look what kind of complexes they make. And what we knew at the outset is that some citrate synthases can make dimers, which you see there at the top, and then some others could make hexamers, which were essentially a trimer of dimers, where the dimers are arranged in a kind of barrel shape. That's what we knew from the outset going in. And this is what their mass spectra look like if you do mass photometry on them, just so you can see what these things kind of look like and what the data quality was. And now we went through this family and we made very many bizarre discoveries as to what kinds of complexes these enzymes could form. And here is just a small selection of them. So we had some very large complexes like this 18-mer here that we're going to talk about in a moment. We had some proteins, it's not very visible here, that could populate multiple stoichiometries at once, just like BSA can, what Justin showed earlier. And in general, what we found is that these enzymes transition incredibly quickly in their stoichiometries. Every 5 to 10 enzymes that we would look at, we would find a new assembly that we hadn't seen before. And what also very often seems to happen is that something evolves a larger assembly, and that larger assembly is then later lost in evolution. And in some cases, we also had sort of a convergent evolution of very similar looking protein complexes. And all of this we could only discover because mass photometry made it so easy for us to purify proteins in relatively small scales and get an accurate measurement of their stoichiometry. But what I want to spend the rest of the talk about is the strangest protein that we found, and that's this 18-mer. And this 18-mer just interested us initially because it's an odd number. It's not obvious how you can make an 18-mer out of a hexamer or a dimer. So I thought this would just be a big old protein ring of dimers with a big hole in the middle. So we decided to image it with electron microscopy. And that's what you see here. This is a class average of this protein where every blob that you see is a dimer. So there are 9 blobs, 18 subunits. And initially, when we got these images, I wasn't really aware of what I was looking at. I just thought it looked kind of amusing because it has this triangular shape. And me personally, I was reminded of the Triforce symbol from the Zelda games. So I sent an image of this to my friend Eric Markland, and I said, look at this hilarious protein Triforce symbol that I found. But he realized that this was something more important. He said, what's interesting about it, and the reason it looks like that Triforce symbol is that it looks like it's a triangle of triangles. So he thought that the corners here, they look like smaller triangles, 3 of which I've arranged into a bigger one. And that reminded him of a famous fractal pattern, the Sierpinski triangle, which is an infinite series of triangles of triangles of triangles and so on. So he asked me when he saw these images if we found any bigger triangles on our EM grids that would be 3 times this thing arranged into an even larger triangle. And sure enough, by the time he asked, we had already found them. And these are these giant 54 subunit triangles with a hole in the middle that's comfortably big enough to fit a ribosome. And I should add that at mass photometry concentrations, these are quite rare. They're maybe 1 to 2% of our, at EM concentrations, they're maybe 1 or 2% of our particles. So these are rare, but they can definitely form. So now we had ourselves we kind of sure at that point that we had a protein Sierpinski triangle. Now, why is this a big deal? And that brings us to what fractals really are. So fractals are structures that retain the same amount of fine structure regardless of how far you zoom in or out of that structure. There are many, so I should say that fractals began in mathematics as a kind of rebellion against calculus, against the idea that you could zoom infinitely into a curve such that you can make it smooth enough to integrate or differentiate. And fractals never do this. No matter how far you zoom in, there's always a lot of fine structure and detail. And we find many structures that have this fractal characteristic in nature. For example, we see this in the branching of riverbeds and river deltas, where you have a big river channel that branches off into intermediate channels, and in a similar branching ratio, those channels then branch off into even smaller things. And that makes it difficult for you to tell at what magnification this image was actually taken at, because it would look the same if I had zoomed in on this river channel here. And similar patterns also form in the drying and cracking of paint, which I was excited to learn as an active area of research in physics. But perhaps the most famous fractal in nature is the Romanesco broccoli. And that's a special fractal because it's regular. In this fractal, the correspondence between higher and lower levels of organization are sort of exact. You have these floral walls that are pyramids, and they're made from smaller pyramids, and those are made from even smaller pyramids. And thus, being a physical fractal, of course, at some point this is going to terminate, and there is a smallest unit. And the Sierpinski triangle, of course, is also a regular fractal because triangles at large scales are made of triangles at smaller scales, and so on. And regular fractals in nature are very rare, and by the time we made our discovery, no regular fractal was known at the molecular scale. This protein was the first molecular fractal known to be formed by molecules. So why was this so unusual? In fact, there were mathematical theories that predicted it shouldn't be easily possible to make regular fractals, specifically Sierpinski triangles, out of molecules. And these theories were essentially based on the rules that we knew proteins usually use to self-assemble into regular structures, into regular-looking structures. And so to understand why these rules can't be used to make a Sierpinski triangle, I just want to quickly go through two very simple requirements that we would need to make one. And we're going to imagine that our smallest building block is this thing here, which would be a hexamer where each triangle is a dimer. We want to make that into a larger triangle. And we need two rules for this. The first rule is that we have to put these little triangles together at the correct external angle of 120 degrees, and that just gives us a bigger triangle. So that one's easy. The second requirement is a bit more subtle. The edges must be passivated such that we can no longer, excuse me, associate any more hexamers into this edge. The only place where I'm allowed to associate more subunits would be the corners. And this is necessary to give rise to the large internal voids of the Sierpinski triangle. So we need these two requirements. So let's think about how we can satisfy them with normal protein-protein interactions. I'm sorry, I'm coughing so much, I'm a bit sick. Okay, see if I can get through this. We're going to try and start to satisfy our angular requirement first. And we can satisfy a 120-degree angle between subunits with a C3 interface. The C terminology comes from the point symmetry groups in crystallography. And these are head-to-tail interfaces. And I've signified this here by indicating that every dimer has one head and one tail. And that gives us the right angle, but you can immediately see the problem because we've satisfied one head and one tail, but each dimer still has one head and one tail open. And that means I can push another hexamer into the edge. And if I propagate that interaction, I get a boring 2D lattice, not a Sierpinski triangle. So let's try a different interaction first that passivates the interface. And that would be a head-to-head interaction. Because most things only have one head, that gets rid of this problem that we can push other subunits into that interaction. And those would be C2 interfaces or isologous interfaces. And the problem with those interactions is that they're made at the wrong angle. And if I propagate this interaction now, I just get another different kind of boring lattice. So something has to give. It's clear that we can't make Sierpinski triangles out of the very normal point group symmetries that we know from thousands of structures in the PDB. So how does it do it? And in order to figure that out, what we had to do is obtain cryo-EM structures of the three fractal levels that we could see. The hexamer, that's the sort of 0th level of the fractal, the 18mer, and the 54mer. And this was very difficult because this was not a well-behaved protein on EM grids. And one of the most annoying problems that this thing caused is that it doesn't like to lie down. What these triangles like to do is stand on their tips such that you kind of see a thin line as if you're looking down at the edge of the triangle. This is what we would most commonly get as a view in the EM grids. So fortunately, Jan Schuller in Marburg is a very fearless cryo-EM microscopist who is willing to extract densities out of even really terrible looking grids, and he succeeded. So the first thing we're going to understand is how we take hexamers and make them into an 18mer. And so we're looking down the edge of the triangle now, and we're looking at two dimers from two different hexamers and how they form that interface. And so ordinarily, in a dimer of dimers, this would be a symmetrical situation where all four chains from the two dimers would participate equally in the interface. And that means they would make exactly the same interactions. And interestingly here, this is not true. So this interface is chiefly held together by an interaction between a histidine and a glutamic acid, between the sort of the blue and the green chain. And you see that in the opposite way down here. But the orange and the pink chain do not participate in that interaction. And the corresponding residues of those chains point into space into such a manner that you couldn't attach another chain here without causing a massive steric clash. So what we've done here is very subtly broken the symmetry of a normal dimer of dimer interactions. And we've done it in such a way that it makes the interaction at the correct angle and that it passivates the interaction, and we can no longer associate other subunits into that edge. So that was step one. And when we got this structure, I thought we were done, because what I thought you now had to do is take this interaction and just propagate it along the edges to take an 18mer and make something even bigger out of it. And when we tried that computationally, it failed. And what you got was a spiral and not a closed triangle. And the reason this happens is that the interaction that we just looked at is made at a very slight tilt. It's not completely planar, but the tilt is small enough that along the distance of an 18mer, you can still force the triangle to close. But if that angle acts out over a longer distance, which you would get if you string three 18mers together, then the two ends of these triangles can no longer meet and the triangle is not closed. So something has to be different. And so for that, we had to solve the structure of the 54mer, the second fractal level. And that was planar. So how does it do it? So it turns out it uses the same surface. So now we're again looking down the edge of the triangle. And the interaction that we're concerned with is right here. And this is the one that we just looked at. So this was the one that holds two hexamers into an 18mer. Here we're holding two 18mers into a 54mer. And it's the same surface, but the dihedral angle between the dimers that make the interaction is very different. In the 18mer is about 60 degrees, and it's roughly half that in the 54mer. And that's necessary such that you can close that triangle. So if you now wanted to take three 54mers and string them into something even larger, that angle would still have to shrink further. And we have indications from our EM grids that this angle can effectively become zero. So this is quite an unusual feature of a protein-protein interaction, because normally the angle of interaction is relatively rigid. So the two things that you kind of needed to make that fractal was the ability to break the symmetry of this interaction of dimers, and then you had to give it enough angular flexibility such that you can close the even larger triangles. Now, the question for us was, is this complicated? Is this something that is so difficult for evolution to build that it needs a function? I mean, clearly these fractals are rare, right? We were the first people to discover a protein-Sierpinski triangle, so there can't be too many of them around. But are they difficult to evolve? And specifically, do they need a function to evolve? And initially, I was convinced this thing had a particular function. And the reason for that was that we noticed by mass photometry that this assembly was very pH sensitive. If you raise the pH, these fractals would fall apart into hexamers. It's a slightly unusual pH change for what's probably mediated by a histidine. So it happens between a pH of about 7 and 9. And that was a suspicious range to us because the organism this comes from, a cyanobacterium, undergoes a strong change in its intracellular pH throughout the day. The pH is relatively basic during the day because it imports bicarbonate, and that's part of its carbon concentration mechanism that it uses to drive CO2 fixation. And then at night, that relaxes back to a normal, sort of more neutral pH. And the other thing that I should say we knew is that the fractal form of the enzyme is less active than the dissociated form. So we thought what happens here is that the enzyme gets shut off at night, because only at night do the fractals form, and when the pH goes higher during the day, they fall apart. And that made sense to us because the Krebs cycle is not only there to produce reducing equivalents that you can then use in metabolism or to drive your electron transport chain, but it's also a source of precursors for nitrogen assimilation in amino acids. And actually, in this cyanobacterium, the circle is not complete. It's a truncated circle, and it's actually mostly used for nitrogen assimilation. And this is a pathway that cyanobacteria shut off at night that was known because it requires a lot of energy at night when they don't have enough energy coming in. So I was convinced this would be why you have to make this fractal. But the problem was that we made mutations that prevented this protein from making the fractals. It could only make the smallest form, the hexamers, which were catalytically still active. And if you did that and you grew this cyanobacterium at different day and night cycles, the mutant never showed a fitness that was different from the wild type. So as best as we can tell, the organism doesn't seem to care whether or not these fractals can be made. Now, I don't know that this protein doesn't have a function in vivo. It's very plausible that we didn't find the right conditions or that our fitness measurements were not sensitive enough, but it doesn't seem to be a strong fitness effect. And at that point, our cyanobacterial collaborators asked us whether we thought it was plausible that this could kind of be a harmless accident of nature. And that's only plausible if this is relatively easy to evolve. If you think about it, your eye as a really complicated structure requires the sort of coordinated expression during your development of thousands of genes. And if any one of them breaks, the eye doesn't form. And something like that doesn't evolve by accident. That requires the deterministic action of natural selection for millions of years, hundreds of millions of years to build that structure. But maybe these complexes are not so complicated, and they might be more easily reachable genetically. So to understand that, we followed the evolution of this fractal assembly. And to do that, we first inferred a phylogenetic tree of the citrate synthase that made this fractal. This is from the Cyanobacterium Synechococcus elongatus, and then all of its relatives from other cyanobacteria. These are the other ones. And if you're not familiar with reading phylogenetic trees, time flows from the root of the tree on the left here into the leaves. So what we then did is we used ancestral sequence reconstruction, and this is a method whereby you can use the tree and the alignment that you created the tree from, together with a model of sequence evolution, to back calculate what the sequence would have been of the protein at the internal nodes of the tree. And these nodes represent ancestral proteins that existed in organisms that are now extinct. And once we have those sequences, we can, of course, just order a gene that codes for them and then resurrect that protein in the lab and characterize it. And so we did that. I'm not going to show the MP data, but we just measured by MP whether they can make these fractals. And what we're going to do now is walk from the organism that makes the fractal into the past, which means towards the root. And the first two nodes, so the youngest two nodes that we encounter, can still make the fractal. And then everything older than that, starting from this orange node, cannot. And then it's very simple. You can then find out what historical mutations were necessary to create this assembly by just comparing the sequences of the orange and the green protein, because the changes in between them must contain those mutations that make the fractal. And this was very bizarre because the only mutation that was anywhere near the interfaces that hold this thing together is this Q to L mutation. And what's strange about that is that these leucines in the fractal are actually too far apart, even though they're right at the fractal interface, but they're too far apart to make a productive interaction across that interface. What we thought might be plausible is that removing the Qs and making them Ls removed a charge repulsion or a polar repulsion between these Qs. And so we tried if introducing this mutation into the orange protein would make the fractal. And it did. So here you see the MP data for this. So here we have the orange protein that just makes hexamers. And if we make the single point mutation, you get a fractal. And if you think about this, this is kind of remarkable because all of the residues that make the positive interactions that hold this thing together, they are all already present in this protein, but for no reason at all. It seems like evolution has brought this protein so close in sequence space to making a fractal that in the end, it could make the jump in a single mutation. And that is as simple genetically as you could imagine. So I'm not going to stand here and tell you that I'm certain this protein evolved by accident, but it certainly has all the qualities necessary to evolve by accident. So one thing I didn't tell you is that, for example, these groups of citrate synthases have all reverted back to making an even simpler assembly. They only make dimers. And there's some other relatives of this protein that I'm not showing on this tree that also quickly lost the ability to make fractals. And the only organism in which this survived is Synechococcus elongatus. Okay, so I'm probably over time. I'm going to skip this slide here. And just to conclude, you can do some pretty funky things just by using the inherent flexibility that proteins have. And if you can make a Sierpinski triangle, who knows what else you can make? These assemblies are not difficult for evolution to build because they exploit features that fortuitously pre-exist. And there's lots of variation in the self-assembly state of even very conserved proteins. And your guess is as good as mine as to why. Some of these probably have functions, others might just come into existence by accident. And I think what's interesting to think about with this fractal is that this only survives in a single organism, and it makes you wonder how many crazy assemblies have come and gone through the eons that we just didn't have the good fortune, or that they just didn't make it into the present for us to see them. And with that, I'm done. I'm sorry if I went a little bit over, and I'm ready for your questions.
Super. Thank you, George. And thank you particularly for fighting through what clearly is a bit of a cold. There are a couple of questions that I wanted to pass on from the audience. The first was sort of musing as to the observation that your fractals are two-dimensional, but proteins often are much more, are not always as planar as that, right?
Yeah.
Could you imagine a way of actually generating Sierpinski pyramids,
Yeah.
with relatively few mutations?
That could definitely be possible. That's a question for the protein designers. The thing that you're fighting there with all fractals is that the concentration jump from one level to the next is really large. So the concentration dependence would be pretty extreme. That in order for these things to not be toxic to the cell, they can't make fractals at really low concentrations because they'd gunk up the cytoplasm if they did. So I'd imagine that would be a significant engineering challenge, but there's no reason why it shouldn't be physically possible.
And from the same question, asking also whether, and I think to some extent you answer this, but asking whether, it says there's an allergy to prion assembly, they feel, but was wondering whether you had an understanding of the fractal assemblies being locked in by a certain fold and self-propagation of that fold. I guess that's the analogy with the prions there.
Yeah, they're not prion-like in the sense that these assemblies are extremely reversible. So you can, just by diluting them, you can get back out of this thing. And we don't see a huge conformational sort of conversion that is irreversible in the same way that it would be in a prion. We have structures of all the levels, and it's, in this case, that's just not how these work.
And someone else asks whether you find these interfaces to be particularly flexible, in the cryo-EM data or any molecular dynamic simulations, whether you've noticed
Yeah, they are quite floppy.
flexibility.
They are quite floppy in the cryo-EM, so the resolution is not great, which is why we had to validate them all with point mutations. And people have now done MD simulations on them and found this angular tolerance in them that they need. And that's not a normal, they're very small, sort of confusingly small interfaces, actually, in terms of the number of residues that participate. And that probably makes that possible.
Okay. And actually, there's one final question. We still have a minute before we move on. Question is asking you, is this similar, if you can draw any analogies with the way carbon arranges, and in graphene and diamond and non-functional fractals there.
Carbon and graphene are still crystalline in the sense that they have translational symmetry across their principal axes. And what's interesting in these fractals is that they always have one threefold symmetry. That's what a Sierpinski triangle always has. But structurally, our fractals otherwise have no translational symmetry, for example, along the edges of the triangle. So they're not like these carbon arrangements or like diamond. And for the question whether these fractals are functional, they're not. So if we lock the protein into this fractal with disulfide bridges, that diminishes the function. So we don't think they're capable of catalysis. They would have to dissociate in order to perform catalysis.
Okay, super. George, thank you very much. And I'd like to invite Maria, if you're able to share your screen. Maria Spies is at the University of Iowa. And it's a real pleasure to have her giving the final talk for today, and talking on really the combination of mass spectrometry with single molecule fluorescence imaging. Over to you, Maria.
Hey, thank you so much for introduction, and it's a really pleasure to be here, and I really enjoyed everyone talk, so I hope you get something out of mine as well. So my lab has been, it's actually our lab logo, so we study the good and the bad of DNA repair, and we look at structures dynamic and do structure-based drug discoveries. So mass photometry has been quite useful for us in the last few years to figure out ideal conditions for structural studies, but also to look at how DNA repair machines form. And today I'm going to talk about part one, poly-ADP ribose polymerase, because that's experiments we did not only to look at binding and assembly, but also at catalysis. Just in case I run out of time, so here's my lab. Most of the work is done by Paris Gaur, so he's here, and also the work was started by former student Fletcher Bain, and Surias helped a lot to Paris with experiments. So why are we looking at DNA repair, and what are we looking at? So the dream of every cell is to become two cells. And in past to this dream, cell experienced a lot of obstacles, because cell has to replicate all its DNA one time per cell cycle, and fatefully under replication, over replication is bad. So, and there are things like DNA damage, difficult to replicate regions in our genomes, and collisions between various machineries working on our DNA. One of the proteins which is a universal sensor of difficult regions and damage is called poly-ADP ribose polymerase. So it's an enzyme which goes around, finds different types of DNA damage, and uses NAD+ to form those long, sometimes branch ADP ribose chains on several types of residues, mostly aspartate glues in proteins, and also can modify nucleic acids. So the arrangement of a protein, so it's about 100 certain kilodalton protein, so it's is arranged like a beads on a string with several functional domains and long linkers between them. When PARP1 binds its the site of DNA damage, it comes together in a kind of more compact structure, and that activates the catalytic site here. So PARP1 has been a very attractive a target for cancer therapy. That is because it is useful in types of cancers which deficient in certain types of DNA repair and have so-called BRCA phenotype. So PARP inhibitors now have been around for an FDA approved in the United States over a decade now, and so these inhibitors are used in various types of cancers, mostly breast, ovarian cancers, and now tested almost in everything which has a deficiency in DNA repair. And that is because it helps clinicians to discriminate between the cells in the patients which are cancerous and which are not. So, but the exact mechanisms, and that would also still kind of interferes with development of a beta new inhibitors, is understanding what exactly is the function of PARP1, which many of its substrates are important, and what should be actually inhibited. So PARP1 is involved in single-strand break recognition and repair. So it processes protein nucleic acid in processing of protein nucleic acid covalent complexes. In several types of DNA repair, it interacts with single-strand breaks, double-strand breaks, stabilizes replication forks. And what we were especially interested in is it recognizes a DNA structure called G quadruplexes. So in our genome, there are over 300,000 sequences which have those 3 Gs separated by 1 to 7 nucleotides, and they're repeated 4 times, or more than 4 times. When in a single-strand form, so these sequences can fold into like knot-like structure, which is stabilized by monovalent cations, by specifically potassium. So these structures are found in human telomeres, in promoters of oncogenes, in promoters of tumor suppressors. They're involved in replication and also can be formed in in repetitive DNA, which is linked to various human diseases. So what we were interested in is to look at how PARP1 interacts with G quadruplexes, both in terms of dynamics, and we wanted to enumerate complexes involving G quadruplexes in PARP1, and to see how commonly used PARP inhibitors affect this complexation. So the 2 experimental techniques which I will be talking about today are a single molecule total internal reflection fluorescent microscopy and mass photometry. Obviously, I'm not going to introduce mass photometry. It's been done fabulously, but with total internal reflection fluorescent microscopy, we also have a surface, except the surface is passivated, and we would like to avoid as much as possible of our proteins sticking to that surface. On the surface, we can attach 1 macromolecule, and it's the most simple experiment here. The other macromolecule is in a solution. We bring in laser light at a very deep angle where, due to the differences in the refractive index between solution and glass or quartz, so it appears, so the light is fully reflected off the surface. So it generates a phenomena called evanescent wave. So it's a wave which propagates along the surface, penetrates our reaction chamber to about not more than 100-ish nanometers. So it decays exponentially. So in a sense, we're only looking at the surface. If we have molecules nailed to the surface, if they're unlabeled, we don't see them. If we have another molecule in solution, we don't see it unless it forms a stable complex with our nailed molecules and persists for at least few frames of our camera. So we know it didn't just float by. In a sense, the experiments are very similar to what Justin described earlier, which he's trying to do with mass photometry. We're looking at kinetics of binding and dissociation. So, okay, now PARP1. So, and from mass photometry part, we wanted to see both interaction with quadruplexes and phosphorylation. By itself, so here is our PARP1. It's about 113 kilodalton. So all perhaps by us and everyone else who works biochemically or structurally with PARP1, we get this extra band around 70 kilodalton. It's a fragment. But in case of mass photometry experiments, we actually use it as also as a marker, an extra weight. So it's about 113 kilodalton. Now, if we want to look at DNA binding, we were looking at 2 substrates. One is a classical DNA substrate, is a single-stranded nick. However, in order to look at binding directly to the nick, we needed to construct DNA substrate, which would not have ends, because PARP1 can also bind to the ends, and if we have double-stranded DNA, we have very complicated assembly of many molecules. So we have a dumbbell where there is no ends, so there is a nick here. In the case of G quadruplex, we have that sequences specifically from promoter over c-KIT gene, so which can form quadruplex, and there are the same dumbbells on the side. So the structure is pretty stable, and also it adds, it's really relatively sizable. We picked up about a balance between what would be distinguishable from the protein and what we can see in mass photometry, and also what would not break our bank to get synthesized. So now if PARP1 undergoes autopyrylation, we were curious whether we will be able to see it, or how large will be the chains and how the protein will interact with the surface in mass photometry experiments. Okay, here is a typical binding experiment. So you saw quite a bit of those today. So we can readily distinguish PARP1 by itself and PARP1 bound to, say, either nick DNA, and we see a shift by an expected molecular weight from 113 to 135 kilodalton, or 252 kilodalton in the case of a quadruplex containing substrate. So now enzymatic activity. So this is done just in a normal buffer. Now we can add NAD, which is a substrate for enzymatic activity. Again, we start with PARP1, we add nicked substrate, we add NAD, and we wait. And so the time here is from when we start a measurement. So, and the measurement is about a minute. So we actually have here representation of about minutes since the marked time here. So in somewhere around 4 to 5 minutes, we grow this large chain. We grow something of a much larger molecular weight. And it's still, surprisingly, it's a very charged polymer, but we get it on our glass surface and it stays. Even larger chains we observed when our PARP1 was bound to quadruplex containing substrate. Actually, if you quantify it, so based on one ribose, so we have so we grow up to 100 kilodalton, 150 kilodalton of those chains. They're variable in length, so you see how wide the distributions are, so of about 200 to 300 units, maybe even more so the tail here. And it's still presumably one protein with a hairy mass of pyrylation on it. So what was also important for us is to see that under substrates like this, we get a binding of one PARP1 per one piece of DNA. Now we can more look at kinetics of binding with a little bit more confidence. However, for this, we need to label our PARP1. So in both cases, so the PARP1 labeled either with size 3 in green here or size 5 in red dye, with about 1 dye per molecule of a protein. And we wanted to confirm that we have this one-to-one binding. So here is a trajectory or time-based change in the fluorescent in a single spot on a slide. Simultaneously, we see anywhere from 200 to 2,000 kind of diffraction separated spots. We see over time things are blinking, but in two colors in this case, and we're looking at one spot at a time, in this case over 400 seconds, and this messy colored events, binding of the red molecule, binding of the green molecule, and then dissociation, binding and dissociation. And in all of our experiments, we never saw molecules of two colors coming at the same time to the same spot. So we can confirm that all of that different flickering of light is just one molecule at a time. So similar to what we see in mass photometry, here we see also one molecule of PARP1 binding to surface nailed quadruplex molecule. And in this case, we can also just to the just to show that our fluorescent labeling, which in this case we put a dye on an N-terminus of protein, and so it does not affect much activity of a protein. We see the same binding. So by mass photometry, we see somewhat significant amount of correlation between experiments. So now, by mass photometry, we were able to see complexes, pretty stable ones, but we needed kinetics, and we went back to quantitatively analyzing our interactions. So one problem with experiments which involve a surface compared to mass photometry, where complexes are in solution and then they land on the surface. Here, one of the molecules, in this case, either DNA or the protein, have to be surface tethered, and that can affect binding. It's actually surprisingly, in this case, it did not, maybe due to flexibility and pretty sizable construct. So, and what we see, so here's an examples of a surface tethered DNA and fluorescent PARP1 in green. So each event here is a binding of a single PARP1 molecule to the surface tethered Nick DNA or c-Kit DNA, and we can measure times between events and times where events are there. Kind of exactly similar to what Justin was showing before with mass photometry kinetics measurements. So we can bin them, the time we can build the histograms, and we can feed them to exponentials. And we see actually numbers which were somewhat similar to what was expected for PARP1 and very similar for both DNA substrates. So we can calculate on rate, off rates, on rate constant, off rate constant, dwell times, and KDs, which happen to be somewhere in the order of 2 to 4 nanomolar. So it's a pretty tight complex, either with a surface tethered PARP1 or surface tethered DNA. So now, oops, sorry. And somehow my things kind of shifted a little bit. So now what happens, so what we couldn't see in mass photometry experiments is whether upon that very significant self-correlation of PARP1, whether the complex stays intact with DNA. So we did an experiment where we added NAD and at the beginning of reaction together with the PARP1. And what we see on various types of substrates, and I'm just showing nicked and quadruplex containing DNA, but if we compare the two parts of a trajectory in the beginning and the end, and again, so these are representative trajectories, so they represent hundreds and hundreds of traces like this. We see only a fraction of trajectories had events in the second half of it. So it can be explained by two things. First of all, correlated PARP1 dissociates from the DNA, or PARP1 can also correlate the DNA. And that is something we're still trying to figure out which is the case. But it's definitely so we don't see the complexes later on. Now, what happens with inhibitors? So one of the first PARP inhibitors is olaparib. So it's inhibitor which prevents PARP1 enzymatic activity, but has not too much, so it has slight so-called trapping effect. So, or enhanced residence times on the DNA in cells. So when we did experiments in the presence of olaparib on unique DNA, so we could see, of course, by itself, it doesn't do anything to the protein, but in the presence of olaparib, our PARP1 forms nice complexes, DNA-bound complexes on unique DNA, but there is no correlation, so we're comparing this sliver and this. So the same happens with two other inhibitors, veliparib and EB47. And again, we saw the same thing with quadruplex containing substrates. So inhibitor works in terms of it prevents pyrrolation, but there is no effect on DNA binding. Of course, we could do all the same experiments and get the kinetics. And what we found was somewhat an interesting thing where on the unique DNA, similar to what people observed in cells, we saw with three inhibitors different effect on KD over PARP4 DNA, and which stem from a longer, a slightly increased residence time, and also changed anon rate. So veliparib is supposed to be actually non-trapping inhibitor, and we see it's pretty much similar complexes as without inhibitor. Olaparib has slight trapping efficiency, so the resident time increases on the substrate, and so is KD is increasing. With EB47, we observed two different types of complexes, long-lived complexes and a little bit shorter-lived complexes, but both were longer-lived and more stable than in the presence of inhibitor. So instead of a single exponential in the bound state, we had this distribution of two exponentials. So we had to recalculate for both of those KDs, and it looks like there is a significant stabilization of the complex, but still complexes are dynamic. And when we used our substrate, which contains G quadruplex, we saw no difference in in on rates, off rates, or apparent KDs. So these inhibitors work differently. So they still affect PARP1 enzymatic activity, but they don't affect the residence time on quadruplex containing substrates. So in conclusion, so we were able to show that PARP1 forms one-to-one complexes similarly on the NICT and G quadruplex containing substrates. So there is a really massive amount of autoporylation which happens on quadruplexes, and there's also preference, as shown before, that for G quadruplexes of parallel topology. And then, so the conundrum we still see is that interactions are pretty short. So we see several seconds of binding, but pyrrolation increases over about 5 minutes in solution. And so we also see the difference in trapping of PARP1 or increasing residence time on NIC DNA, but not on G quadruplex containing DNA. And again, so these are people who are doing work, so people who are paying our bills, and I think I'm in time to answer a few questions.
Thank you, Maria. Really lovely talk. And indeed, there are some questions for you. So if we start with the first one, which asks you whether you think it's in principle possible to actually more directly combine the mass photometry and the TERF, do that almost within a single instrument to see both outputs in real time.
So I think it's more of a question for you than for me, because so we built our TERF instruments, but I don't see why not. I think it could be a really good a future for this technology. It may be Actually, it would be a combination because our turf microscope is using prism to do excitation, and so we collect the light using objectives, so it could be consistent with.
Yeah, I think it's an engineering challenge, not a physics challenge, right?
It's yes, yes.
Another question here was asking, how you went about actually measuring the DNA samples. And talked about whether you had to treat your sample. Sorry, you had to treat your slides to do so. The question said they've had some challenges themselves when using poly-L lysine, for instance. So any tips or tricks there would be helpful, I think.
So in this case, we tried to compare regular slides with polylysine slides, and we like just the regular slides. Depending upon the type of DNA experiments, we cannot trust. So we can trust molecular weights in a complex. DNA sometimes shows somewhere we expected, but we can see a shift in normal glass slides, which correspond to the mass of DNA, but we're just looking at the protein. And it doesn't work for every protein. So we had a lot of hard times with some other proteins, and complexes were just fine. We never try, so I don't see it now, but so very large complexes, I'm not sure, so we would need much larger DNA because of the
And
noise.
I'm not sure if you mentioned it, but if you look just at the DNA on its own, did you have to use special glass slides for that, or did you not do that?
We just see less counts, but the ones we see are close overall, slightly shifted from where we expected. So for this around 30 kilodalton. For a lot of other experiments, we use about 8 to 10 kilodalton molecules. So those we don't see. So these are at the borderline. So we see something, but again, so the counts are much less than we expect them to be.
Mm-hmm.
And also for DNA, we see both binding and dissociation. We always see, so for larger DNA molecules, which we can estimate the mass, like we have replication forks, which are about 70 kilodalton, and we see identical symmetric peaks on both sides of the zeros. So we see on and off.
Okay, great. George, do you have a question for
Yeah,
Maria?
great talk, Maria. I just wanted to ask, when you did the MP under the reaction conditions and you saw this distribution shift, I guess because you're adding all these modifications on?
Yeah, so they're happening while in solution.
Yeah, I was just wondering, so are you are you very confident that adding these modifications doesn't also change the binding? So do you think that you can trust this mass shift? Because I'm asking because we've had lots of problems with that sort of thing. That's so I'm wondering if you have a trick.
No, it's not a trick. We just don't so we quantify the mass. We don't quantify the number of counts. And actually, in this experiment, you can see the reduction in overall number of counts because we have that contaminating entity of 70 kilodalton, and that thing stands the same. But even if you, of course, our distribution became much broader, but if you count all the counts there, there's still a little bit less.
Oh, interesting.
And we start also seeing dissociation.
Yeah, very cool. Fascinating. Thank you.
Thanks.
Okay, I think that's answered all the questions that have come in. So thank you very much, Maria. And before we enter our discussion, let me just thank George and Sarah again for I think combined to really a really great series of talks. And I hope this have highlighted to listeners, how people are using mass photometry in that protein science context. So thank you to all of you. We have about 10 minutes left as scheduled to pick up on some themes that maybe were raised in the questions or other things we'd like to discuss. So I suggest we do that. And I guess the first question or point that I wanted just to put up for discussion really is about this idea of, mass sensitivity and whether there are problems, whether it's sensitivity to shape and size of these molecules. Is that a confounding factor or something to be, essentially considered as negligible? And maybe, George, I don't know what your feeling is here, right? But you had some proteins that are pretty unusually, pretty unusual aspect ratios, right?
Yeah, I should say that we are still even with the bigger fractals, we're still pretty far under these size limitations that you quoted, because I think, yeah, it's like 100 angstrom across for the 18 or 200 angstrom or something. It's not huge. So we're still far under the 200 nanometers. But I think one thing that is a bigger problem for us, that these very large assemblies, if you make them out of any protein, are typically very concentration sensitive. Maybe not for viral capsids, but for other things. And we've had a very hard time capturing things that we see on EM grids at decent concentrations. We had a hard time seeing that on the MP. And I still have no good explanation for this, because we've tried everything. We've tried the sort of high concentration modifications to the MP, and just could never see that stuff. But on occasion, you would see one or two of these very big things, and they still had normal looking diffraction limited spots. So to me, that's not a huge, the scattering size is not really a particularly limiting factor. I think it's more that maybe very large things don't like binding to surfaces, because there's too much accumulated charge. I don't know.
Interesting. Have either of you, Sarah and Maria, found certain things that to be undetectable, or if that's the right word? I mean, you mentioned a bit about DNA, Maria, that obviously when things get very small, they fall below the mass range of MP. But have you got any other sort of comments based on what George is describing?
I think sorry, go ahead.
Maria.
So there are several proteins we struggled with in this, which form filaments on DNA. So from all other data, including cryo and other biophysical experiments, we can see that they form complex. But in solution, they might exist in monomers, dimers, trimers, and they show this distribution of species. And we cannot force them, or we cannot see the actual complexes we expect. We know that they should be there, but we have no idea where they go, and it's possible that what we see is just complexes which don't interact with the surface well enough.
Sarah, what was you had something to add?
In my experience, we've had a few cases where it was more an issue of the concentration being too low for the complexes to form with the technique. But for us, it's more, especially when working with membrane proteins, it's more of a challenge when we're working with the a mass range that falls within the mass of the micelles, where we're very hard time being able to distinguish what is coming from the buffer. And that's in my experience, that's more been my challenge, I would say.
What's that?
I think I have one more comment to this. I mean, because there are so many biochemists potentially in the audience, we do sort of a lot of service work for cryo-EM data sets. And it's very commonly the case that we have to cross-link to make complexes visible that they can still see in cryo-EM grids. And they often come to us also to see if the cross-linking sort of perturbs the structure that they think they're going to get. But that is pretty common. So there's quite a lot of biological complexes, I think, that are probably not strong enough under our mild buffer conditions to hold together. And I'm not really sure what's missing, to be honest, what holds them together, if it's just crowding in the cell. But
yeah, I think that's right. I mean, so certainly, you could do a similar cross-linking experiment and do, as you said, George, and try and see whether those oligomeric states do appear or do persist. I suppose an interesting question is that, with in a mass photometry experiment, you have a certain number of molecules landing at a certain rate, right? And you're going to measure for a certain amount of time. So you have a certain number of events, and you should be able, in principle, to detect rare events because as long as you wait long enough, right? And how many events do you feel you need in order to be able to confidently assign those events as being something that you actually want to interpret confidently. So the signal has to come from somewhere. You don't just get something appearing, at 2 megadaltons that wasn't anything at all. But people, I think, often want a little bit more than that. And I guess in the cryo-EM example, George, you can hunt around on a grid and find one thing that looks like you expect it to, and it's automatically validated by the fact that it looks like what you expect it to look like. Whereas it's perhaps more difficult or more challenging in our situation where you just get one readout and there's no sort of additional validation of that. I don't know how many molecules instinctively you feel you need to be able to say confidently that I've detected it.
It's a good question. I think you would probably be challenged by reviewers if the fraction is too low. Like even if you have a nice peak because you've done 5 hours of MP on the same sample, and that you have 500 counts, but if they're out of 500,000 counts, I'm not sure that would fly past peer review. But you're right, it has to come from somewhere. I think because I think this came up in the questions to your talk, Justin. I think a bigger concern also has been that you see unexpected stoichiometries, things that people don't think should be the functional form of the enzyme. We've had that these enzymes make multiple oligomers at the same time, much like BSA does. We had to experimentally prove that those stoichiometries are functional because people thought they were just aggregates. And I just think that I think biology is messier in terms of the assemblies that it makes than what we're used to from crystallography, or even from classification by cryo-EM. And we just have to accept that stoichiometries are not as neat as structural biology would have you think.
And I think, we all know the crystallography and these things, as you said, you introduce bias in some of those measurements as well, right? If you're doing classification cryo-EM, you choose to work on a subset of your images. If you do crystallography, not everything will crystallize, right? And you're enhancing for what will form that packing. So, I don't think we're artifactually generating stoichiometries that don't exist in the mass photomics, right? I don't think that's what you were saying. I think it just maybe opens our eyes sometimes to, as you said, the messiness.
Actually, for the messiness, so I see 2 questions in the chat about proteins with IDRs or unfolded disordered proteins.
Yeah.
And the answers there, I guess.
I'm happy to have a first answer of those. A question, or way of thinking about this, right, is when you measure your diffraction limited volume, you're looking at everything in there and seeing how what you're looking at is within that volume, how much of that volume is water and how much is something else. So typically protein, right? So that's how the refractive index of that volume is going to change. And so whether the protein is folded or extended, it's still displacing the same amount of water ultimately, right? So you expect the same signal for an IDP of a certain mass as you do a globular protein of a certain mass. Within the caveats that I mentioned, that once you start to get, into over 100 nanometers and approaching 200 nanometers, that things might start to get a little bit funky looking. But I think that for the IDPs that we're being asked here by the questioners, that will almost never apply, I think. I don't know if any of you have an extra comment on that.
If I can, because we've worked with the mass spectrometry in denaturing conditions, and I guess it would depend. Maybe there is a bias depending on the mass range that we're working with, but in our experience, we haven't seen any significance of mass measurements working in denaturing conditions, and especially because We have implemented it for cross-linking as a way to measure the complexation rates of our assemblies. And we use mass photometry in denaturing conditions to be able to optimize the condition of cross-linking for MS studies, but can probably also be applied for cryo-EM if you want to stabilize a complex and stuff like this.
That's great. It's a really good point. And it's also worth remembering, right, when people talk about a protein being unfolded, we often mentally have this image of something being this elongated sort of string. But of course, random coil is actually still a pretty globular object in a sense, right? Proteins don't suddenly, because they're unfolded, become long extended chains. Perhaps I think just because we're basically at time, but I think there's an interesting question maybe to finish up on, and I think Maria, you can answer this well, which is a question asking about sort of being able to observe rare events and being able to monitor rapid transient interactions. So the question asked specifically about protein kinases binding substrates and releasing phosphorylated products. Do we think it's possible, to imagine a situation where MP could be used to actually watch these transient encounter complexes and reactions and so on, doing things in real time?
I think it's possible, but it depends upon the system and concentrations, because you want to measure. So you sample for, say, at least a minute, and if and your binding and phosphorylation and release happens in solution. So for anything with just rare binding and dissociation, you can have that fraction of molecules because you're looking at a snapshot what's out there. But if your reaction is happening, so you're, I think you will see something. But the question is, it's similar to what Georg said earlier. So what? So how many rare events would be considered real by reviewers? And if a lot of them are happening in solution before you start recording them when they are dumped on the surface, so you may miss a lot of those.
Yeah, and that's right. And I think one of the things that I'm optimistic about the methodology that I introduced is the ability to continuously monitor a single molecule for a length of time, and thereby watch the state changes that it undergoes. And by doing that over thousands of molecules to actually get meaningful statistics and mechanistic information is ultimately the aim.
But
especially if you can surface tether the kinase, and you will see its substrates coming and going.
I think what we would have to be mindful about is the residence times, because we have the integration time of the MP experiment. We have to acquire multiple images to average it together enough, which the instrument in some ways hides from you in the software how it's doing this. So we've tried it with very transient interactions, and we've really struggled to see them. Like we knew that under the conditions where we did the MP, we had encounter complexes forming and so on, but you have to change the sampling rate of the camera all the way down and so on. Your resolution goes down. So that, I think, I have nothing to do with instrument development, but to me that there still seems to be a technical challenge there. That many of these reactions are along the time scale that the instrument would actually struggle to see, because you have some integration time.
Yeah, I think that's right. And I think there are trade-offs there. It would be easier to see something on a bigger system than a small system and so on. But that's certainly something that needs to be taken into account. I think we're a little bit past time, but I really value you all sharing your thoughts. So thank you again to you. I think we'd like to thank Raluca and Shannon and the Protein Society in general for supporting the talks today and the topic in general. And I think with that, we can draw the session to a close and hope you will be able to join the next Protein Society webinar that sounded really quite interesting when it was introduced. Thank you very much and have a nice day.
Thank you.
Thank you, everyone.
Bye-bye.
Bye.